Hello guys, I'm Nurul Aqilah and my friends here is.
My name is Nabilah.
And so today we're both here to summarize the new finding about copy number variance CNV. So let us proceed to the video.
Based on the news excerpt titled Novel CNVs Uncovered Affecting Genes Involved in Drug Metabolism, it has been found that copy number variants CNV contribute to variability in genes linked to drug metabolism. A copy number variation CNV is DNA segment of 1 kilobase or larger that is present at a variable copy number in comparison with a reference genome. A team of international researchers had conducted a study in which they collect data from 1000 Genomes Project and the Exome Aggregation Consortium. The study was conducted to examine CNVs affecting more than 200 known pharmacogenes. As for the result, they have found that novel deletions and duplications in most of the genes they look at. They have also uncovered novel CNVs significantly contribute to the functionality of relevant pharmacogenes, adding an additional layer of pharmacogenetic complexity with important implications for the prediction of drug response and toxicity. Using the same data, the researchers conducted a bioinformatics analysis of 208 genes involved in drug pharmacokinetics. They have found that 97% of those harbored novel CNVs. In total, they identified 5,589 novel CNVs, and of those, slightly less than half were deletions and slightly more than half were duplications. They determined the minor allele frequencies of the CNVs in certain human populations from this data. Most of the deletions the researchers uncovered were population-specific. The most common deletions they found affected cytochrome P2C19 in fins with an MAF of 1.1%, cytochrome P2B6 in Africans with a MAF of 0.9%, and cytochrome P4F2 in East Asians with a MAF of 0.4%. The most frequent exonic duplications, meanwhile, affected CBR3 and cytochrome P2B6 with MAFs of 0.7% in fins and 0.4% in Africans respectively. Rodriguez-Antena and her colleagues confirmed a subset of the novel deletions they uncovered using Sanger sequencing, and further performed CNV genotyping of 3 genes, including cytochrome P2C19, cytochrome P4F2 and SLC01B3 in 1,080 people from Spain, 465 people from Finland and 590 people from Japan. They found that the cytochrome P2C19 exon 1 to 5 deletion had a MAF of 0.43% in the Finnish cohort. Cytochrome P2C19 is involved in the metabolism of a number of drugs and is included in the dosing guidelines of 21 US Food and Drug Administration drug labels, including labels for citalopram, clobazam, clopidogrel, escitalopram, and flibanserin. Full cytochrome P4F2 gene deletion, meanwhile, had a MAF of 1.61% in the Japanese cohort, higher than the East Asian MAF of 0.36%, which the researchers said could reflect a population-specific distribution of the deletion. They also added that they found one Japanese individual who was homozygous for the deletion. Additionally, the SLC01B3 exon 9 to 13 deletion had a MAF of 0.53% in the Spanish cohort. SLC01B3 encodes OATP1B3, which the researchers said helps clear bilirubin and is involved in the transport of other drugs. The MAF of this deletion is some 5 times higher in Spanish individuals than in Finns, which they attributed to population-specific differences. In conclusion, Rodriguez-Antena and her colleagues conclude that folding in CNV data could help determine which pharmacotherapies might work best for certain patients.
So that is all for our sharing session today. So thank you for watching.